Hello students, welcome to Module 6.1.1, Phylum Chordata. Of course, this is a phylum that includes the vertebrates, animals with backbones. This is also the first module after our last lecture exam, exam 5. The material that is presented here will be tested on the final exam. The final exam will be half comprehensive material from previous exams and half non-comprehensive, and this material will make up the non-comprehensive part of the final exam. I would also point out that all of the lectures for this non-comprehensive material are presently in one single large PowerPoint. In the future I might break that up, but for now it's all in that one PowerPoint presentation. So check that out. But the modules will be in individual little chunks as the past ones have been. Of course, now we come to the last phylum of the semester, which is perhaps a good thing. Here we can see animals with backbones. These are the animals that are most familiar to most people, but what is not familiar to most people is the phylum in which they are classified. And so vertebrates are animals with backbones, this other major dichotomy in the animal kingdom. Previously we looked at invertebrates, animals without backbones. Now we're going to look at those with backbones. The thing about vertebrates is that they are classified into phylum Chordata. Now this phylum has very diverse members, and so we're going to look at 4 features which unite all of the different animals that are classified into this phylum into that grouping. The first feature that we're going to consider is the dorsal hollow nerve cord, and this is of course an extension of the brain that goes down and is protected in the backbone, in the vertebral column. So here where I'm running the cursor over, these are the vertebra which make up the vertebral column, and what you can see coming out of here are spinal nerves, branches of the dorsal hollow nerve cord, or the spinal cord. The spinal cord is the dorsal hollow nerve cord. It's called dorsal because dorsal means more toward the back than the front. The front is ventral, and a good way to remember this is think of a V-neck sweater, and the word ventral starts with V, and so that's the V goes in the front. Dorsal is in the back. Think of a dorsal fin of a shark. In any case, in the body of an animal, vertebrate, the nerve cord is more dorsally located. It is also hollow. If you look at the bottom left illustration here, this is a transverse or cross section through dorsal hollow nerve cord, or through the spinal cord, and you can see that little white spot right there. That's the hollow, and you kind of get the idea. There's this H shape in the middle, and on the outside we have white matter, and the H shape is the gray matter. It's kind of interesting because it reverses onto the brain. The gray matter's on the outside, the white matter's on the inside. But in any case, this is a nerve cord, dorsal hollow nerve cord. That is not the feature for which phylum Chordata is named. Notice a difference in spelling of cord, C-O-R-D, and chordata, C-H-O-R-D-A-T-A. Here is the feature for which Chordata is named. It is the notochord. This is a rod which runs along the long axis of the vertebrate body. It has a supportive function. In some aquatic forms, like fishes, it can be like a torsion spring. So the fish would use muscles to bend the tail to one side, and then the notochord would cause it to snap back like a spring. Notice that I put here it's made out of cartilage, but then I put a question mark. Some people have questioned that it's made out of cartilage like other cartilage, but it is some cartilaginous type or like substance that makes up this feature. Now, it is present in the adults of some fishes, but it becomes much reduced to just intervertebral discs in many vertebrates, including mammals, reptiles, birds. And so as a supportive element, it is replaced in these groups by the vertebral column. This little illustration here, this shows an embryo of a frog the African clawed frog Xenopus, and you can see the eye spot right there. And it has been, this embryo has been especially stained to show the notochord, and so you can see the staining of the notochord running along the long axis of the body. And this is the eye spot up here in the front. And notice the spelling, cord and chordata. And so chordates have notochords, at least at some stage in their development. A third feature which unites members of phylum Chordata are pharyngeal gill slits. And pharyngeal comes from pharynx. The pharynx is the back of the throat, the area where in you both food and air pass. So pharyngeal gill slits. Gill slits are openings. Here we can see in the lower left the red oval labeled pharyngeal gill slits. These are present in a jawless fish, a lamprey. Here, this is a human embryo, and we can see gill slits. The gill slits do not persist in the human embryo. Rather, they become other structures, ears and nostrils and terrestrial forms. In fishes, these slits are used to have water run over the gills, and therefore they allow respiration or exchange of respiratory gases. But in other forms, they become other structures, as noted. The fourth unifying feature of phylum Chordata is a postanal tail. We can abbreviate that PAT, not to be confused with point after touchdown, but this is a tail that is present after the anus. In some forms, here we can see in the upper left, a kangaroo, it can be used as a counterbalance. In aquatic forms, it's used for propulsion. A fish has often a broad fin on its tail, known as a caudal fin, and this allows it to swim. But here's also a gray squirrel used as a counterbalance in that group as well. I think these are all humans on the right here. Several human embryos showing tails. The tail is typically reabsorbed, but here we can see two examples in which it was not reabsorbed. And tails vary. Some of them in humans, some of them are just little fleshy things. Some of them have an extension of vertebra in them. But what happens typically in westernized hospitals is babies born with tails, they're simply snipped off. What is the normal developmental pattern in humans is that before birth the tail is reabsorbed, but in some cases that doesn't happen, and those are humans that are born with tails. Here we can see a kind of conglomerate. This is a hypothetical chordate, and it shows all four of the major chordate features. The dorsal hollow nerve cord, the notochord, the pharyngeal gill slits, or pharyngeal slits, they simply label them here, and the postanal tail. So those are the four features that members of this phylum have. Under phylum Chordata, the last phylum of the semester, because this is the last one and this is the one to which we belong, we're going to study it in more detail. So we're going to look at subphyla. Phyla, of course, is a plural of phylums. There's no such thing as phylums, so we say phyla. So the three subphyla under phylum Chordata are subphylum Urochordata, subphylum Cephalochordata, and subphylum Vertebrata. Two of these are invertebrates, subphylum Urochordata and subphylum Cephalochordata. Sometimes we will call these first two subphyla protochordates. They are invertebrates. These are proto means first, the first chordates, and these are invertebrates. This is the only phylum to contain both invertebrates and vertebrates, is phylum Chordata. So I don't know what else we can make those letters do, but you should remember that fact. We're going to quickly look at the protochordates, and then we'll move on and look at subphylum Vertebrata, which is where we'll spend most of our time. But we do want you to be introduced to these first two groups. So first let's look at subphylum Urochordata. And if we look at all of the taxonomy here, of course, this is kingdom Animalia, phylum Chordata, subphylum Urochordata. The etymology of Urochordata, Euro means tail in chordata means chordate. So these are the tail chordates. But the common names, that CN here stands for common names, of these animals are tunicates or sea squirts. All these animals are aquatic, and they live all in salt water, so they're all marine. These animals are filter feeders. They have an immature form called a tadpole larva, and they undergo a retrograde metamorphosis. Retrograde means going backwards. The mobile larva has all four chordate features. It has pharyngeal gill slits, and then in its tail it has the dorsal hollow nerve cord, the notochord, and it has the tail itself. But this swimming tadpole larva, which is microscopic, in lab we will see one, but it's on a microscope slide, so they're pretty small. The mobile larva has all four of those chordate features, but it settles down on its head and it reabsorbs its tail. And so it loses three of the four chordate features and retains only the pharyngeal gill slits as an adult. And this is what the adult form looks like. Kind of looks almost like a kettle. An interesting fact is that this covering or tunic, can you think what a tunic is? In terms of clothing, it might be something from the Middle Ages or something, and it's kind of like a robe or a covering. And the tunic here is made out of cellulose, the same thing that the cell walls of plant cells are made out of, but that's convergent evolution, because we don't think that it has this tunic made out of cellulose because it's related to plants, but it is an example of using glucose as a monomer in two different groups. And so here we can see the cellulose tunic. This is actually a little drawing here on the right of a microscopic larva, and it shows all four of the chordate features you can see in white boxes: dorsal hollow nerve cord, notochord, and tail, and then the pharynx with slits or pharyngeal gill slits. And here we can see after it's settled down that it's lost three of the four and retained only the pharyngeal gill slits. Here's actually a photograph of a microscopic view of a tadpole larva. And here's some beautiful blue tunicates, and here's one in a jar. Sometimes I say that it looks like someone sneezed in a plastic bag, the plastic bag being the tunic or covering, and some really difficult to discern stuff inside. But you can see here's a stylized drawing that interprets kind of the anatomy of these animals. They are filter feeders for the most part as well. And with that, we're going to leave subphylum Urochordata and talk about subphylum Cephalochordata, the second subphylum of invertebrates found in phylum Chordata. In this case, cephalo means head, so these are the head chordates. The common name of these organisms are lancelets. A lance is like a spear, it has a linear body, and LET is a diminutive, so it means like a little lance. These guys are about an inch or two long, and they're all marine. They tend to bury their tails in the substrate, the sand or whatever, and their heads are poking out, and they filter feed. These animals are segmented, and here we can see the segmentation in the form of these muscle fibers or myotomes that we see. And we can also see the many pharyngeal gill slits. Notice that there are many. And the adults retain all four of the chordate features, and we can see an interpretive drawing here. This is showing them buried. This is a dead one out of a jar, like the ones we have in lab, that someone stuck in an aquarium. And it shows the real thing. It really shows the muscle fibers or the segmentation here. The segmentation in vertebrates is separately derived than the segmentation in the earthworms and the arthropods. So the four features there are dorsal hollow nerve cord, notochord, pharyngeal gill slits, and the tail. And here it asks the question, this is showing a slide, we'll have such a slide displayed in lab. It says phylum, subphylum. And so the phylum is Chordata, the subphylum is Cephalochordata, the common name is lancelets. And with that, we're going to conclude this introductory module.